Welcome to Life Science Solutions. I'm your host, Chris Adkins, and today I'm joined by Andrea Arsiccio, a senior scientist and team lead in Silico at Coriolis Pharma. And he joins me today for a deep dive on in silico modeling usage in drug development, especially as it applies to assessing developability and the ability to develop a reasonable formulation for new drug products. So, Andrea, welcome to the show.
Thank you very much for this great opportunity. Very happy to be here.
Yeah. So it's really, really good to have you, and appreciate you, being here in RTP with us today. Maybe before we deep dive into the in silico aspect, and I'm really excited and curious for that. Some of our audience probably has some familiarity with Coriolis Pharma, but I'm sure some do not. Can you describe a little bit about where Coriolis, kind of fits in the life science ecosystem, and specifically what makes Coriolis unique in the drug development landscape?
Yeah, sure. So at Coriolis is a contract research and development organization. So we are service providers, and we have a lot of experience in drug product development, with a clear focus on the development of formulations for liquid products, frozen liquids, lyo products. And we can also support with a huge variety of analytics, both in R&D and GMP. Mm-hmm. And I would say what is special, what is unique about Coriolis is our focus on science and our ability to innovate. For instance, I am part of this team at Coriolis. It's called Science and Technology. So what we work on is really development of new techniques, research. We write scientific articles, so we contribute to really scientific literature. Yeah. And it's a big focus for us. And I would say also what is also unique is the high level of experience and expertise. So we have, for instance, a scientific advisory board composed of professors from also the US, for instance, that support us in quite complex and challenging projects with their expertise in the field, and also our management. They have a huge amount of collective years of experience in pharma services. Mm-hmm. And I would say as well is like we really deal with challenging molecules, so that's where we have a lot of experience.
Yeah. You're not working on the simple formulations.
No, we don't receive them. Yeah, that's the thing. Yes.
Yeah. Kind of reminds me of a moniker we hear internally here a lot, where we'll question like, well, why don't we get any of the easy programs to work on? And it's like, well, the easy ones are those are all easy and they're already done, right?
Correct. Yeah. That's the point.
Yeah. Very good. So you lead the In Silico team at Coriolis. Can you I think this is probably a term that a lot of folks have heard, for most of us, myself included, kind of, before this conversation, more in passing. But can you talk about and explain what does in silico modeling mean in the context of drug development? And why is it becoming, maybe this ties into our last comment, but why is it becoming increasingly important in biologics and advanced therapies?
Yeah, that's a very good point. So, in silico refers to whatever is done on a computer. So whatever type of calculation is performed to support, for instance, in our case, the development of drug products. And it comprises a lot of different type of models, like mechanistic models, computational fluid dynamics, for instance, heat and mass balance equations, physics-based models. I'm thinking about molecular simulations, molecular dynamics, molecular docking, and obviously also what is becoming more and more popular, artificial intelligence, like machine learning, so data-driven models. The reason why it is becoming more and more important to apply in silico in our development products is because we see the value of in silico applications to speed up the drug development process, and also to de-risk this process. So to make like in keywords, better drugs and faster. So that's the objective at the end of the story, and how it supports drug development process. For instance, in drug discovery, if I think about molecular simulations, molecular docking, molecular dynamics, and as well also artificial intelligence, or like clinical studies, reduce, for instance, the number of animal-based experiments pharmacokinetic, pharmacodynamic, toxicologic studies. In silico applications are like widespread there. Or even AI to like something simpler to draft or write applications for submissions to authorities. So that's another field where AI is becoming more and more relevant.
Yeah. And it's really kind of across the board, it's started to boil to the top, not just because of all the movement in AI, or maybe it is because of it that we see in the economy as a whole. But now, of course, we see, the FDA saying, hey, we're going to use AI for application review, right? We've already implemented that. That's underway, which is fascinating. The rate of change, a little terrifying as well for anyone who's used AI, more than 5 minutes, right? Yeah. But then also the expectation that, hey, we really expect that the level of animal testing required be driven down, right? Yeah. And it's going to be through the use of techniques, right, like this to assess risk, because ultimately, right now, we default to animal toxicology because it's the best approximation we have to not impact, hurt, harm, kill otherwise healthy human beings. So that's the importance of kind of really developing these types of technologies really well, and then also there's a whole preclinical development piece that you touched on, right, and the efficiencies that you can gain there, whereas before.
Yeah, you can increase potency, you can increase affinity, specificity, reduce immunogenicity by just modeling, not even performing experiments in the lab. And that's
it's like the crude explanation to me is, the original way to kind of test if you had a XY grid of 5 by 5 different options for whether it's formulation or what have you, that you would have to test 25 different options, right? Yeah, well, then we evolved and we said, okay, well, we're going to do design of experiments. We're going to bring that testing load down. Some of it is we got some kind of innovative, different. I'm thinking especially from like the upstream process development perspective. There's some innovative technology that lets you test more variables more easily. Some equipment out there, but this seems like the next step for that, right?
Correct. Yes.
Yeah. So and it kind of reminds me of the digital twins on the manufacturing side have become really popular to talk about and even implement, because you can assess change before you do it. So this seems like almost the development analogy to that.
Absolutely. And AI also is used extensively to even suggest manufacturing conditions, so even in combination with digital twins. So
yeah. So maybe taking a quick step back, how did you end up in the in silico modeling world, right? I mean, you're a classically trained chemical engineer. Yeah. So how did you end up here? And do you think that what you do now is more computer science or more chemical engineering or something else?
I think it's something in between, I would say. So I try still to stick also to what I learned during my chemical engineering studies. But that's a very good question. So since I was a child, I always wanted to give my contribution to supporting the health of people, right? At the same time, I had a passion for maths, physics. So I thought about starting engineering, and specifically chemical engineering, because it still allowed me to select courses that were focusing on pharma processes, pharma manufacturing, and so on. And back then, I focused mostly on mechanistic approaches, so heat and mass balance equations, the hard core of a chemical engineer. Computational fluid dynamics as well, so very physics-based models. I then moved during already my PhD into molecular simulations, and I started applying them extensively. So molecular dynamics, molecular dockings, also a lot of bioinformatics, thermodynamic modeling, and so on. And in recent years, I found out how important and how the potential of artificial intelligence and how it can really support traditional models as well. And it's how I then transitioned into this in silico world. So it was a progress.
Yeah.
It was a long journey.
Yeah. I can imagine. I'm curious, and I don't know. I don't think we'll have time to get into it, but I have to imagine that the state of technology today makes both the potential impact of the models so much higher, but also I would expect the burden to create them. So I did some research in undergrad on kind of one-dimensional particle modeling. And that was just because we weren't going to go to 2 or 3 dimensions because our model was already far too cumbersome to make, right? And computers have come a long ways in 15 years as well. But still, I think to what it would mean to implement that today, and just the leveraging of AI for the coding purposes.
Yes. That makes coding so much faster.
Yes,
Yeah, it's impressive.
yes.
And I agree with you. I don't consider myself old, but since when I started doing my PhD, like computational power has changed so massively. Calculation that back in the days took like 1 week or 1 month. Now you can complete them in a few days. So it's really a big change. It's.
I mean, I'll never forget. I mean, I built my first desktop, right? And switching from compiling my code on my laptop to on my desktop, what would take 16 hours now took, I mean, like literally 20 minutes, right? So anyways. So kind of specifically, I mean, with that application of AI, how does it apply to the current modeling work that you guys are working on, and really, where does that get into, again, your biologics and your advanced therapies? Where does it get into accelerating the formulation development and kind of de-risking some of those?
Yeah. We use AI and in general in silico applications for mainly 2 purposes. So on the one end, we use it for candidate screening. So when we receive molecules, we try to use machine learning models, for instance, so AI, to identify liabilities. So is a molecule sensitive to, chemical degradation, conformational instability, colloidal instability? That's the question that we try to answer with our models. At the same time, as you mentioned, like Coriolis is a strong focus on formulation development. So what I think is also unique is the way we apply in silico tools and artificial intelligence to also suggesting formulation corridors for a given molecule. So we don't rely only on identification of liabilities, but also on solutions, how to deal with these liabilities, how to formulate these molecules in such a way that they can become successful drug products.
Okay. So looking at it from the kind of the total picture.
Correct.
Right?
Correct.
Would be a differentiator, because you can leverage all of your historic formulation knowledge.
That's correct.
Right? Because you guys, it's not like Coriolis came into existence 2 years ago or something like that, right? I don't remember offhand how old the company is, but
yes, more than 15 years.
Exactly.
Yeah, so
exactly.
we have quite some expertise in developing formulations. Yes.
Yeah. I guess maybe to kind of peek in a little bit more. I mean, so you guys have kind of this systematic antibody preformulation platform that has been built out, right? Can you talk a little bit about what makes that platform unique, and how does it really drive that selection of a best lead formulation earlier in development?
Yeah, exactly. So what we try to do is to combine our in silico approaches with wet lab. We have a lot of experience in wet lab experimentation. So on the one end, when we receive a new molecule, we apply our in silico tools and we combine them with developability assessments in the lab. So we test conformational stability. We test colloidal stability. We measure hydrophobicity, for instance. So we try to characterize the molecule also from a wet lab perspective. Yeah. And as you mentioned, we also combine our in silico with pre-formulation screening. The unique part of that is that we apply a lot of high throughput techniques with low volume consuming methods, so we can test a huge number of formulations in a relatively short time. Yeah. And when we do this high throughput screening, we combine, as already mentioned, quite a lot of techniques. So we try to characterize a molecule quite extensively. We test again conformational stability. We do nanodifferential scanning fluorimetry, for instance. We test formation of particles. Yeah. We do both soluble particles like size exclusion chromatography and subvisible particles. And we test also chemical degradation via ion exchange chromatography. And we can do that without consuming a lot of material. As you know, especially in the beginning, it's very hard, it's very challenging to produce a lot of material for a new molecule.
Yes, and especially in the advanced therapy space.
Correct.
Right?
Correct.
Yeah.
And that's where these high throughput, low volume consuming techniques become extremely relevant.
Yeah, so having that wet lab expertise to back up the in silico, right?
Yes, they go hand in hand in the end to thoroughly characterize
because it's
candidates.
tough whenever you see folks that are going to come into this space and they're going to do some of this in silico work because they've developed models and they're going to do things, but none of that wet lab, experience exists, right? I mean, it's analogous in our world to if you're going to do device design and development, right, from a delivery system perspective, but then you can't put it on a bench and test it.
Yes.
Right?
Yes.
Like if you can't 3D print, buy your molded parts, whatever it's going to be, and then put some force profile data together, you haven't proven anything until you've done it in the real world.
That's
right?
True.
It's like the pinnacle of the scientific method. Like you have to actually do the thing, right? So
makes sense.
Yeah. Can you talk about maybe a specific example where this in silico modeling has helped solve whether it's a formulation or a process challenge? I mean, you hit on some of the big ones, right? Particulate formulation, other compatibility issues, and something that would have, presumably been a lot harder to solve without using the tool.
Yeah, I think a big advantage of in silico tools is that you can really unveil the mechanism behind, for instance, some instabilities you observe in the lab, right? So we had a case, for instance, of a molecule that was showing a lot of aggregation, and it was very hard to formulate. And as you know, formulation is also some sort of art. And to make it become science, you need to understand what is the mechanism behind what you observe in the lab,
Not
right?
just what's happening, but why is it happening?
Why it is happening, right?
Yeah, which is really hard. Yeah.
Correct. And by applying our in silico tools and comparing, for instance, these molecules against databases of approved molecules, we could find some in silico outlying properties that were really pinpointing to a given mechanism of aggregation, And when you find this out, you know how to address it. So you know how to solve this problem. So that was really
because you know why it's
correct.
happening.
You know why it is happening, and you know how to fight this problem with your formulation. So that's, I think, where in silico has a lot of potential. So another example, what is very common for us is to receive clients to support clients that have a lot of molecules in their pipeline at the very beginning, like preclinical phase, and they want to select a given lead candidate, you know? And if you wanted to do that with only wet lab, you would need to produce maybe a lot of molecules, and it would cost a lot of money. So if you can already pre-screen this huge amount of candidates via in silico, that is a huge impact on the cost of drug product development, for instance.
Yeah. I want to circle back to that, the comment, not only what is happening, but why is it happening? And I think about, I started my career in early phase development. We made preclinical and phase 1 clinical trial material. But even just the impact of that, knowing why it's happening, and then and the trickle through to your process, right? Because you don't just know, okay, well, we changed this and it solved the problem, but I don't know, was it a temperature, was it a mixed time, was it a hold time?
Yes.
And when I understand why, I can then have that impact my critical process parameters as I'm building out my kind of technology understanding of my entire platform.
That's correct. Mechanistic understanding is key, like for any type of, as you mentioned, not only formulation development, but processing, manufacturing. You really need to know how your molecule behaves.
Yeah, exactly, right? Because and in those early phases, that data is one, precious, but two, is very, very limited.
Yes.
Right?
Yes.
It actually makes the quality aspect in that early phase manufacturing is it's easy to think that maybe it would be lessened because you're in early phase clinical and you're not held to a commercial standard. But in my opinion, it's actually much harder because you don't know what the baseline should be all the time, because there are unknowns in your process, right? You don't have a validated scale down model. You don't have 10 years of manufacturing experience on it, right? So driving to that what is going on would be so beneficial from an early development, and ultimately you would be, I think, in the truest form following QBD principles.
That's
what
we try to do
out. Yeah. So, how do you do that blending of computational and experimental data? Like how do you manage the handoff between data simulation, hands-on lab validation? Like what's that kind of look like at Coriolis?
Yes, our vision is that in silico tools and wet lab validation go hand in hand, and especially at the beginning when we start developing a new model, we try to validate as extensively as possible. So we need to prove that the model is credible, and we need to prove its extrapolation capabilities as well. That's crucial. When we have proven credibility and extrapolation capabilities of a model, then we try to also use the model itself to reduce the number of wet lab experiments you need to perform, to focus experiments. That in the end has to be in our vision, or in my opinion, the objective of in silico applications as well, right? To speed up the process by the knowledge that you gain via the in silico tools.
Yeah. I'm curious, as you're implementing these, and I think it's one thing if you come at a problem, that a client has from like quite early phase, like you said, someone who has a list of targets and they're trying to figure out, I use the which lead candidate are they going to hitch their wagon to, right? Because especially if you're a small startup company, like so much of your lifeblood just matters how that lead candidate performs. But when someone comes to you and they already have some aspects of their quality target product profile, or maybe they've already done some development work and they already know some CQAs, how do you implement those knowledges that they've gained? And I'm going to guess they probably gained them the hard way most of the time, right?
Yeah,
they've done they've done a bunch of experimentation and proved what doesn't work or what really makes the molecule unhappy. How do you implement those into the AI-driven workflows to again leverage that knowledge that the client is coming up with, and also reap the benefits of the in silico model?
Yes, as you were mentioning before, quality by design, right? And that's indeed our vision. And to apply quality by design, it is important to include information about the QTTP as early as possible. And for instance, a way we do that is based on some QTTP specifications is to focus on some liabilities of the molecule. For instance, and I want to give an example. Now there is increased demand of subcutaneous applications. And therefore, instance, it becomes, in many cases, extremely important, for instance, to work at high concentration, right? Concentration of a mAb or of a protein in general. And for these type of molecules, for instance, sometimes a problem that you face during formulation development is viscosity. For high concentration.
Gets very, very high, very, very fast.
Right. And that's where we also can then, for instance, focus with our in silico tools, focus on solutions to address viscosity or estimation of excipients to reduce viscosity. So that's one example.
Yeah. And shameless plug, we had a whole 2-part series, episodes on ultra high concentration drug formulation and the different options that exist there, whether from a delivery or from a formulation perspective. So I think that kind of leads us into, obviously it can be really good to generate this data, and it gives you a lot of really good scientific input and helps you validate some of what you're going to do from a wet chemistry or even then from an ongoing process development perspective. From a regulatory perspective, though, how are the agencies currently viewing, AI and the data that comes out of these in silico models? Are you seeing more openness to including some of this in lieu of whether it's demonstrated process development work or even getting into other experimentation? I mean, we talked about tox earlier, but just kind of broad perspective. What's your perspective?
Yeah. I think that agencies are really seeing the potential of in silico tools and artificial intelligence, for instance, to support drug product development. So agencies started releasing, for instance, draft guidances. The FDA released a draft guidance on the application of AI in drug product development. I think it was early this year. The EMA also in Europe released, I think, a reflection paper in autumn and fall last year. And what all of these guidances focus on is how to assess credibility of the model, how to verify and validate this model. And the main conclusion is that this verification and validation activities should be proportional to the risk of the model itself. So how it influences decision and what is the risk associated in taking decisions based on the model, right? So I think this is a very, it's a crucial aspect indeed. So that's where agencies are very careful, and it makes a lot of sense. You need to ensure credibility. And I think with these guidances in place, clarify the needed verification and validation steps, we're making a big step forward.
Yeah. Yeah, and I mean, really, I don't think that any of that should be a surprise to anyone that the agency would always want a risk-based approach, and we only see more and more of that, and some of it is kind of old hat to us because it's been so much more prevalent in the device world than it has in the drug world, right? But we see it more and more. We see it in the EU ATMP guidance when we talk about potency assay development, things like that. Some of the harder assays to build, and especially when you have a limited product and limited data set. But just being risk aware and making decisions in that capacity, right? Continues to be more and more of what the agency expects, and it's so much of what we bring to drug development companies on a regular basis, right? Is the really coherent way to do that. So I'm curious kind of about Coriolis kind of as a whole. You guys recently announced an expansion into the U.S., right here into RTP. So that's super exciting. Just a few minutes drive from where we're at now. I'm curious, kind of both from the physical brick and mortar perspective, but then also from the digital, and whether it's from the modeling or other scientific innovations, right? Where do you see things kind of going from here for Coriolis in those two regards, and what are you most excited about?
Yes, we are based in Munich, right? So and for us, now opening this new site here close to you as well, it's a big milestone. So we are very happy about this. It's a great opportunity. We are proud of being part of this welcoming community, and thanks actually also for hosting me here today. And our aim is to provide better and faster service, for instance, to North American clients now with opening of this new site, and also solving some hurdles that come from also shipping samples.
Sure. There's some logistical
to Europe,
benefits,
right? So some logistic aspects. So what we will start doing, for instance, here in the US, starting already from Q1 2026, is to work on our established and streamlined particle ID workflow, where we combine SEMIDX, Raman, FTIR to troubleshooting particle formation and formulations. And then we'll move down with use studies and application of analytics for drug product characterization. And then later on during the year, so still planning for 2026, is to move to liquid and lyoformulation development. But that's our idea at the moment. But obviously, if there are, we learned that it's important to listen to the needs of the market. And if there are any suggestions out there, please feel free to reach out to us. We are happy to listen.
Yeah. And you guys have been really engaged, right? And we've enjoyed all the conversations so far. So maybe last question. I'm curious, how do you really see on the AI-driven kind of in silico approaches to drug product development? How do you see it evolving over the next few years? And what do you think the impact from that is going to be on biologics and advanced therapies as a whole?
Yes. I think what we've been also working on is digitalization, right? I think it will make a big impact. In the end, when you develop these data-driven models, you need data. And we focused a lot on working on smarter use of data to developing better, more accurate models. We will work also on the expansion of these models to different modalities. Working in silico tools for monoclonal antibodies, rather easy. There are data sets, but we're expanding to harder modalities, protein internal peptides, nucleic acid therapeutics. It's a field that where with Coriolis we will focus quite a lot in the next years. We will do research on those. We will try to contribute to scientific innovations in the field. We're also working on in silico applications to process development. With a colleague that is actually US-based, Rick Van Bilmont, we work quite a lot on development of models for lyophilization, for instance, to support lyoprocess development, lyoprocess transfer. So there is a lot of innovation going on in there, and I'm really looking forward to that.
Yeah. Yeah, well, it's certainly exciting times. The field certainly needs it, right? And we look forward to seeing some of those benefits as opposed to just hearing about it whenever there are problems in formulation and folks are trying to solve those so they can continue to move forward through development. So. Well, excellent. Well, Andrea, really appreciate the time. Appreciate you being all the way here. All the way here in RTP and taking some time while you're here in the U.S. to sit down and chat with us. Really enjoyed the chat. Looking forward to having you guys here in RTP permanently and to more conversations to come.
We're looking forward as well.
Yeah. So thanks so much, and thanks to everyone for listening.
Thanks.
Thanks for tuning in to Life Science Solutions, presented by Chimenox and produced by WalkWest. For more insights like this, subscribe and follow Life Science Solutions wherever you get your podcast. We'll see you next time as we continue exploring what it takes to bring life science innovation to life, because patients deserve better.